I got started in this because my son was very ill with Bartonella for many, many years. So I have firsthand experience with a lot of this, and I wish I didn't, but I do. So just wanted to tell you that. All right, and that's how I got into it. I got into this whole thing. I was studying ehrlichiosis and anaplasma with a friend in the entomology department, and my specialty is using high-end microscopes. And so I got some nice images, and then my son was sick, and then everything's just steamrolled after that. And I'm still doing it. Like this is about year 17 now. So, okay, so I'm going to talk to you a little bit about some recent advances in the role of biofilms in vector-borne diseases. This you guys may know it, but this is our friend the deer tick. This is Ixodes scapularis, and this is the picture that kind of got me involved. This is a nice shot, and this is the hypostome, just different. I just have it in different colors, and it's in the ear of a hamster, and that was how we were looking at an animal model when we were doing that. So I'm going to talk about biofilms. I love this picture. I just found this on the internet, and this is a scanning EM, so you can see all the three-dimensional shapes, and they've pseudo-colored so all these yellow guys here. These are all painted the same, but they have pseudo-colored all the different organisms in this feces sample with a different color, and it's kind of like ugh, it's kind of like oh my. So I'm going to talk about how biofilms play an important role and a not yet well understood role in vector-borne diseases. And by that, I'm going to primarily talk about Bartonella, but I will also talk about Borrelia, and I really won't talk about Babesia, but I like to think of them as the big three. This is technical, but I'm going to try and just keep it basic so we can all understand it. So what is a biofilm? This is a famous picture if you were studying biofilms. So here's some bacteria here, number one, and they all have their little flagella on them. They're flying all over the place. And then they get stuck on a surface of some kind, or maybe not even a surface. And then they lose their flagella and they become kind of stationary, and they keep growing and growing, and they form this biofilm matrix outside of them. I think of it as their house. And then if for some reason the bacteria inside become awakened, they're kind of asleep here. If they become awakened, they can then break free from that biofilm and do their nasty work, or in some cases good work. And what I hope you can see is what it says here. Bacteria living in a biofilm can exhibit a 10 to 1,000-fold increase in antibiotic resistance compared to bacteria in the planktonic, and this is the planktonic state. But they can be free floating as well. And I don't want to spend too much on that, but we'll keep going. Okay, so right now, and I told you I'm going to focus on Bartonella. We know biofilms play a role in bartonellosis, and we know that anti-biofilm treatment is recommended. But the question is, what evidence do we have of Bartonella biofilms in humans? Well, not a whole lot, but it's definitely increasing. And how do we know that any biofilm disrupting treatments, oops, I got it twice, or treatment approaches are proof of efficacy? How do we know they're actually breaking down biofilms? That's a tricky one. So what things I want to talk about today, again, this is Ixodes scapularis, again, taken with one of my $1 million microscopes, and it's not mine, it belongs to the school. So first question I want to talk about, where are these pathogens found in humans? Where have we actually found them? And what techniques do we use to find them or detect them? Then where have we actually found Bartonella biofilms? And why they're important? And then what kind of treatments we can postulate might work. And I have some Easter eggs, if I'm using that term correctly, that I'm going to talk about. This picture on the left is actually Bartonella that we grew in culture. It's just teeny weeny. And so where have we found Bartonella and Borrelia? So anything these are all the places where we found Bartonella. It's a lot more than just blood. And anything that's underlined is what we've done in my lab. And everything else is, oh wait, I can do heart. Yeah, we can do that too. The imaging techniques we use have allowed us to visualize these bacteria in all these different organs. And where has the biofilm been found? We know for sure heart valves. That's due to some elegant work by a friend of mine in Berlin. And we know blood. I've been focusing a lot of my work in the dermis of the skin, the upper layers, because I was in dermatology for many years. And where else? Well, we're still working on that. Okay, I'm not going to talk too much about this, except I want to just notice there's a whole bunch of different ways, different types of microscopy that we can use to visualize these organisms, none of which are trivial. I will tell you that. In fact, here's one. This is what's called super resolution confocal microscopy. Okay, essentially what we've done, we've used a marker that will just highlight the green that's outside of the red cell, and the red is inside the red cell. And you can see this is loaded with Bartonella. All right, and then so how do we see them? Well, we use different colored dyes or fluorescent reporters. And again, I'm not going to go into it, except we look at all kinds of different things. We look at DNA, RNA, structural proteins, all kinds of things. And we can use specific ones for DNA, antibodies, etc. Those are our targets. This is our probes, and then we have our reporters. So I just show you this to show how complex it is, and again, how many fabulous tools we have. So I showed you those fancy pictures of the fluorescent ticks. And how does that work? Well, that's because we use these probes that are specific to different, if you think of this as a rainbow, just laid flat. So we use lasers that will excite just this part of the rainbow, or just this one, or this one, or this one. So it allows us to be very specific in what we can visualize. All right, so here's just a kind of complex overhaul, but this is what we do. So if we want to detect tissues in pathogen in tissues or blood, so this is you or me, we'll take a skin biopsy, and that skin biopsy then gets stained and processed so that we can use advanced microscopy like something like this, and we'll talk about that one later. Or we might just draw blood, which I'm sure you probably all had, and there's different ways to look at blood too. We can look at the RNA. That's some of the machinery that's responsible for the growth of the pathogen. Or we can also just do some very high-end molecular analysis. So I talked about this before. So and I did, this is a nice picture of FISH. FISH stands for fluorescence in situ hybridization. That means we put a probe that's fluorescent green, and we incubate the red cells with it, and we can get these probes into the red cell, and then we take, nice microscopy images, and we can get really nice images that show that these bacteria are definitely inside the red cell. So because I worked in dermatology, again, I have a nice skin pictures, actually a lot of them. So this was actually from my son. His lower leg just did a skin, and he had no therapy yet. And then I used different probes and different reporters to look at it. So the blue is the blood vessels. The green is any kind of a gram-negative bacteria, and the red is Bartonella. And you can see that it's in these little spots. And if you take this picture apart, here's all the red, here's all the gram-negative, and here they are together. So the point of this is he has a lot of gram-negative bacteria, and they were much more than just Bartonella henselae, which, as it turned out, because we worked with Dr. Breitschvert, he's has Galaxy Labs. He's a veterinarian out in North Carolina, real good friend. And he was the one who discovered Hansen's. At least he had at least three different strains of Bartonella. So one of the things I said I would talk about is, where do we find these? Well, this is a little scary, but I've actually found them. It's kind of hard to see, but the red here is nerves. That's a marker we use for nerves. And this is a marker we use for Bartonella. And we can see, if you just look at this little string here and here, and we're just starting to do this work, but it's really great because, as you know, people that have these diseases have a lot of neuropathy, a lot of pain. So we're going to try, and that's one of the things we're looking at. So we were actually lucky enough to get a cadaver from a gentleman who was infected many years. And it was such a generous gift. And one of the things we can use this cadaver for, the cadaver skin that we have, it can be used as just a beautiful, positive specimen for us. So this is incredible. This is the nerves in this arm. So we're doing some studies on nerves, as I indicated. This is just our very first one, and we're going to do better. But the main point is, see all these blue dots here. This is the upper surface of your skin, the epidermis, and down below that you see all these little red dots. These are the Bartonella. So, and this is just how it looks in skin we've taken from people that are, live. And so this was a wonderful gift, and it's going to help a lot of researchers. So I said I would talk mostly about Bartonella, but I will talk about Borrelia as well. So this is Borrelia burgdorferi. This was a surprise. This was a skin biopsy from a woman's abdomen, and we stained it so we could look at the blood vessels. That's here. We could look at green, which is the Borrelia. That's the Lyme disease. And you can see that here. And the purple is the DNA. So we want to know, is this just a nothing, or is it something? So that's why we always put a lot of different probes on. And the green and the magenta together, when they're all put together, they make white. So this is actually, this shows a spirochete transitioning to a ribbon spirochete. So Borrelia is not just a spirochete. It can be a coccoid, a little ball, or it can be a ribbon, or anything in between. But we've not seen very many spirochetes in tissue. So this was enlightening for us. Then some more Bartonella. So this is skin. This is that same skin biopsy. Here's the epidermis. So we've just kind of cut it long ways. And it shows, here's the boundary. This red is, here's the epidermis, and here's the dermis. And so we were looking for Bartonella, and I told you it's teeny weeny. So how are we going to find it? But if you look close here, you can see some, and this is just, this kind of dull green is, we just call it endogenous fluorescence, because it's just what makes up the skin. And if you look closer though, this red is a blood vessel, and all these green dots are Bartonella. And they're kind of, I think of them as like little, I tell you, I have given this lecture to people and they like, oh, what? Because everybody says it's in here. No, it's not. It's all over the place, and that's why that's important. So this is Bartonella in the dermis, and it's some are in the blood vessel, most are not. So here's what I want to tell you about this, because it's so dang important, and I'll keep working in the biofilm, because that plays an important role here. So here we found Bartonella in malignant melanoma. And this was because our good friend Paul Reicheter, now in Kansas, he was at one time in Oregon, and he's a Mohs surgeon, so he takes skin cancer lesions off. And he had a lot of patients who had metastatic melanoma lesions in non-sun exposed areas. Well, what the heck? So he talks to our friend Ed Reichford. Sorry, I have a lung thing. Anyway, and then Ed starts doing his stuff, and then they call me and they say, Marna, can you look at some biopsies? And I said, sure. So, and I think I don't have to tell you guys how deadly, especially when it's unidentified, undiagnosed and undetected, malignant melanoma can be. So we had 8 patients, and we're just submitting a grant to try and get money so we can do more. And these are the different Bartonellas that they looked for. Now they did some antibody titers. This is Ed did this stuff. So here's Vinsonii, Hensley, San Antonio 2, and Bartonella cholerae. So, and what we find when they're in black, we say it's positive because it has a certain level. So essentially it says we had 5 of them with positive serology, 3 of them, and you'll see that 2 of them are up here, or 1's up there, had positive PCR, and only 1 didn't have positive. Something I want to drive home is that if you're positive by serology, that doesn't mean PCR is going to pick it up. Doesn't mean immunostaining is going to pick it up. This is a huge problem, and we're trying to figure out why can't we see these in all the tests, but you can't. So if you have a negative by serology, is it there? And if you have a positive by PCR, yeah, I would go get treated for sure, because not all protocols can pick these up. So we use this kind of the same technique here, and we took lesional skin from 2 patients that was really good, and then we stained it. And I'll walk you through this quickly, so it's because I know it's a pain to look at. So we stained it with a marker in white. This is just to say we've got DNA, so that's good. This green one says you have Bartonella. This one says you have this VEGF. This is very important for cancer, vascular endothelial growth factor. And that encourages the growth of blood vessels, which is the last thing you want when you've got melanoma. And then we also look at lymphatics because they often move amongst the lymphatics. So in this one, we put the green and the white together and showing that, yes, we do have, it's always good to check. I always check. Yes, the Bartonella has DNA, so yeah, I trust it. Then I put Bartonella and the VEGF-C. Well, you can see there's virtually no green. It's yellow, and that's those 2 colors together. That tells us that the Bartonella is inside the vascular system. No, I'm sorry, it doesn't. This tells us that that Bartonella is producing VEGF-C, which it does. This one tells us that it's inside the vascular system, which is inside the lymphatics, and that's the bacteria that are in there. So this is an invaluable picture for us. So then we do what most people do to try and make it easier. We did a co-culture. We used organisms that were just Bartonella. They're in green here, and the DNA is actually tumor cells, metastatic melanoma tumor cells, and we mix them together, and then we look to see how do they interact. And one of the very surprising things, well, 2 of them, they started growing these long processes. I think you can see them and that's like a melanoma cell, metastatic melanoma would do. But also we found that there was Bartonella inside the melanoma cells. Never been seen before. And so this is a 3D picture, and I'm going to show this to you. And this is just a model. This is done with a model system. And I'm just showing you that we found this, the green is now, those are the melanoma cells, and the red is the Bartonella. And the Bartonella is definitely inside and dividing inside these melanoma cells. Because there's a lot of ways you could say, it's not doing anything, but yeah, it is doing something. So this is an important contribution to understanding. All right. And just quickly, what they have found, bacteria-derived, these are immune peptides in melanoma. That's important because it's telling us that pieces of the bacteria are presented to the tumor cells and they change the immune response. In other words, and in the case of Bartonella, it tells our immune system, nothing's going on here, just leave us alone, there's no infection. And we also found Borrelia in those same lesions. We often find Borrelia and Bartonella together. And the green here is the Borrelia, but now you can see it's in its spheroid or coxoid form. The white is the DNA and the red is the lymphatics. And this is within the patient's metastatic melanoma lesion. Just wanted to show you that some other folks took it further, and they actually gave people that had melanoma, they gave them antibiotics, and they found that they were associated with reduced risk of progression. So that's actually very important. Anything that could take it from, so it doesn't go to stage 4 would be fabulous. And then I have some other friends in, these are the Japanese folks, and they found that there's all kinds of special signaling factors produced by Bartonella that influence angiogenesis, blood vessel growth. And here's an example of Bartonella bacilliformis. That's the ancient version of Bartonella, and it's also called arroyo fever and Peru Viragono. I've got the names, I can't remember right now. Anyway, it gives you these external blood lesions that are formed with the Bartonella bacilliformis, and this was how it was first identified, was from Dr. Barton down in Peru. This is important because it's telling us that here's just a visual cue that you're getting a lot of angiogenesis. We found it, I just want to show a couple more things. We found it in appendix. This is an appendix tissue. That the green is the Bartonella, and these are the red blood cells going in a vessel. We have found it, these are all things we've published. This one was with Dr. Breitschwert, and we found it in dog heart. This was a really hard one to do. We found Bartonella in bone. This was from another veterinarian who was having hip resection, so she donated her hip to us after she got new hips and started on antibiotics. And these little white things are the Bartonella. They're abundant in the cartilage. This is from a young man who had, I think you've seen these things. If you know much about Bartonella, then some people will call these stria, and I hate that name because it doesn't really tell us, it tells you there's a stria, and most dermatologists, when they see that, they will say, oh, you've got stretch marks. You've been taking steroids or you've been lifting weights. Nope, that is not it. So we took biopsies from adjacent tissue and from at the leading edge of these things. And then we did the staining with the Bartonella probes. So when I say immunoreactive, that means it has an antibody against Bartonella Hansley. And we did find plenty in adjacent tissue, but we found even more in the scar tissue, which I call Bartonella tracts. And this is the collagen that we found. This is just the collagen. This is a signal collagen gives in a special kind of imaging called SHG. And this shows the scar tissue that's formed. And that's just another picture of that, just a little bit bigger. This is normal collagen. This is abnormal collagen in the Bartonella tract. And we went further with that. And this I did with a friend in Brazil. And so I told you to get these pictures, we just use a special microscope that picks up just the signal from the collagen. So we did. And this is skin tissue again. And we've just pseudo-colored it. We made it magenta. You can make it any color you want. And the green, these are the same. Only this is just the Bartonella. And we find that it is laying along the Bartonella, along the collagen fibrils. And that's kind of what people are finding, is it tends to do that. And then it forms a biofilm. So can we find biofilm in skin? Why, yes, we can. There it is. We also found Bartonella in brain. This is from a little gal that for 8 years, they thought she had a glioblastoma, but it ended up she had vasculitis, a cerebral infarction from Bartonella in her brain. And finally, again, talking with my buddy Ed, we got things worked out for her. We also received some samples from patients that took their life, and this is brain tissue from gentlemen. And the green is the Bartonella, and this shows the Bartonella, and the purple is the nuclear dye. So it's quite abundant in his brain tissue. He had a lot of mental health issues. And we took that same tissue and we also stained it for Borrelia. And that's been found in the brain. And this is the red, this is the blood vessels. And you can see that it's in brain tissue as well. So where we found biofilm, I talked a little bit about the dermis. I'm going to talk about heart valves and blood. This is a colleague in Florida who works with Bartonella. He made a synthetic scaffold, then he put a bunch of Bartonella on top of it, and they waited for it to grow, and it grew this dense biofilm. And he did a lot of work on looking at that. It was really nice work. So we also grew some Bartonella, just Bartonella, in culture, and this is what we got. This is the SEM picture. So this shows the biofilm that's growing. And this is another view, and this is a 3D image, and it shows more of the Bartonella in this thick biofilm. This is a PICC line that's just full of goo. And the white is the Bartonella in there, and the red is actually this one, this pinky one is biofilm. So there's Bartonella in the biofilm inside the catheter that was delivering the medication to kill the Bartonella. And this is a different picture, and these little white balls here, I need to color them. They are Bartonella. It's the same picture, but this is a section, and this is a scanning EM. And what you'll see, I always try and show these more than one way, just to make sure I know what I'm looking at. This is a great picture of Bartonella biofilm. Like I said, it's an aggregate. It can grow on a lot of different surfaces. If you have a boat, you just look on the outside, you'll see that nice biofilm on the edge where the water hits the boat. They've been around for 3.5 billion years, and they are embedded in what's called an EPS, extracellular polymeric substance. And that substance comes from the Bartonella, and it comes from the host as well. This is a blood smear of a patient with Bartonella, and we stained it with some different probes, the green, that would show biofilms in a blood smear. And yes, indeed it did. And this is purposely dirty because I didn't want to do much with it. And these pink ones in here are the Bartonella. All right, then this is again more, we just grew it on different Petri dishes. And we did it also with fish staining as well. And that's looking at the ribosomal RNA. If you've got RNA, you know that this stuff is dividing. That's the beauty of doing fish. This is another nice picture, just a typical stained blood smear you'd get at the doctor's office. But if you look on the edges, you can find these areas of just goo, and they're all over, and that's biofilm in the blood. Here's a great picture by Wagner et al. Sorry, I didn't put the reference down, but the red here is, so this is a vessel, and this red gunk is all the biofilm lining it. So you wonder why you're sick? I'm not. This is another, we published this paper. This is the Buffy coat. When you spin blood down, I don't know if you've ever done it, it gets kind of, this is all the white cells essentially. And so we found a lot of DNAs in the green and in the blue, and all this green stuff is the dividing Bartonella. So just again, another way to show it. This is just biofilm that grew on another blood smear. And this is a human heart valve. This is not an uncommon thing. This is from Annette Moller in Germany, and she has perfected a really great technique. These are heart valves that have been removed because they need to be replaced. And these people were infected with Bartonella quintana, also known as trench fever, which is what J.R.R. Tolkien had and why he had to leave the trenches during, also called trench fever. Anyway, she has done an elegant job to look at. The purple is just all the bacteria, and the yellow stuff is the Bartonella. And so we have also been doing some SEM. This is just to show you we can do it. We had to show ourselves that we can do it. But these are so great. This is MRSA, and you all probably know about that. You don't want it, but this shows the intricate interconnectedness of the biofilm. And we are also using new markers called zDNA. Not that makes so much difference, but I think you can see that we've got Bartonella, and this is skin on mouse skin. So what do you do about it? Well, I've been working with a fellow, Vince Rotello, out in, where is he? I get the wrong institution. He's out east. It'll come to me. Anyway, he designs particles that will get through biofilms, because a lot of times you can have, like, this is triclosan, antibiotic, or we use like eugenol. That's an essential oil that's antimicrobial. But he designs this whole particle. He makes these things, and that because of how they're made, with the positive charges here, they can actually get into the biofilm, and when they get in there, then they can kill the bacteria. And whereas if you don't have that covering, you can't do it. Now, people even have shown that you can get some people have shown, I don't know if I quite agree with it, that bacteria or antibiotics can go in and out of biofilms. Well, maybe, because here's the point. If they're sleeping, they're not going to be susceptible. They have to somehow be awakened, and by breaking up the biofilm, that wakes up the bacteria, and then they become susceptible. So this guy's genius. He's really good. He's at MIT. That's where he is. So, and these are a couple of those little Easter eggs I was going to tell you about, because these are just so important new articles. They aren't necessarily, they are and they aren't. Everything's related to the biofilm, but these aren't. So this talks about, they found Bartonella DNA, and they looked at, this is in Brazil, they looked at bags of blood from 500 so-called healthy blood donors, and 23% of them carried Bartonella. This is not good. This is higher than the amount of Babesia that is used, and that is a cutoff for getting your blood thrown out of the Red Cross up here. We actually met with the Red Cross, but we didn't have the data then. So this is not good. There's a lot of Bartonella, and what's really bad about this is that means when you get if you get a blood transfusion, and say you're an immunosuppressed patient that's getting a kidney transplant, you're going to be in a jam if you get this blood. We kind of did some studies on that too. Also, that same group did another one where they found patients. I talked about that Bartonella bacilliformis, the one that's in the.
This is a new thing for him. This is
a continual pattern thing.
Yeah, this, but like Mom said, this is how he is, unfortunately.
Okay, so
And
should I do Selena's guardianship paperwork? Just to get it done.
who's going to be the guard?
I did quite was that for me?
No, I just muted him. Sorry. It was
okay.
just
All
logged
right.
on. Yeah,
That's fine.
sorry about that.
So and the reason this is important is that these patients with the royal fever, they've got Bartonella bacilliformis, and it was stored at 4 degrees, but they could still find it at 30 months. The point of this is these guys hang around for a long time. And then again, Dr. Maggi and Brightsford did another study recently, just in 23. And at that time, they found that if they could dry out the Bartonella henselae, because it is viable in cat feces, or in, not cat feces, maybe it is, but in the feces of fleas that are on the cats that bite you. Anyway, it's viable. So they did a study to see if they put the Bartonella into biological and non-biological fluids, like milk or serum or something, and they found that they were viable, and even though they were desiccated, that means dried out, they were still stable. I'm just trying to show everybody there really is, these guys are well equipped at sticking around. All right, so now I'm getting towards the end here, and I want to show you this, I think, and I showed you this. This is the Bartonella biofilm on collagen in your skin. Okay, if you have fillers put in, this is what you get, some of this purple stuff. And the important part about this is kind of leads us to having everything I said. Here's the cartoon figure we come up with. So this white box is skin. This is a blood vessel. It's got special cells on the outside, and it's got all the red cells in the middle that flow around, and this is a heart. These purple things are collagen fibrils. All right, so you get bit by this tick or flea or sandfly or spider. There's a lot of organisms that can carry it. And that's when you initially get infected. But what happens is, number 2 here, is that these bacteria send out signals that say to us, we're the host, they'd say, no, there's nothing here. Don't pay any attention. So that means our body doesn't really put up much of an immune response, a fight to this bacteria. And one of the ways we know that this will grow is by growing blood vessels. And then we talked a little bit about that. And here's the collagen fibrils. Well, here, I just use the blue here as the biofilm. And this can serve as the reservoir, like where the quiet Bartonella is. And that's why I say I haven't found a piece of tissue yet where I can't find it if someone's infected, which is scary. But some decide, okay, we're not gonna stay, we're gonna go. And so they'll enter the vascular system, and some may go to the heart and cause a lot of trouble. So someone's gonna need a heart transplant. They'll go to the brain, they'll go to capillaries, they'll go to lymph nodes, etc. It's a very prolific organism. So what's our hypotheses? What are we testing? So we believe that Bartonella biofilms, especially those ones on the collagen fibrils, these provide a little niche, a little home for the persisters. These are the bacteria that persist, and there they are. Our second hypothesis is that the dermis is really the primary niche. This is the primary home for the sleeping bacteria, the sleeping Bartonella. And I have, there's other much more intricate scientific works have done that kind of showed that as well. These are just some of our pals that we've worked with. Dr. Bemis up in Duluth, and Andrea and Zach in her lab did a lot of work at the University of Minnesota, very elegant imaging center. Working with my folks at Tlab. Dr. Maluki is a dermatologist who's from Iraq and is very savvy about these diseases. Dr. Mozieni, he's a brilliant rheumatologist. We've worked closely with Dr. Radovsky and Asani Stoddard in St. Paul and other folks too. I talked about Amherst. I talked about Vince Rotello. Dr. Breitschwert and Maggie are in North Carolina, and Paul's in Kansas. And I've left some people off, but these are just kind of some of the work we've talked about. So here's another lovely picture of a deer tick. And what I want you just to notice, you see these little things on the side? When these guys go bite you, okay, it's hard to pull back. Well, why is it? Because it's dug its little claws into you, and it's got little side claws to stable it. It's amazing to learn how well equipped these organisms are at transmitting diseases. Okay, I'm done. That's it. Any questions? Or I know it's a bit much sometimes, but if you think of questions or have anything, what I've really shown you is a cutting edge of science on these things.
I have a
So,
question.
yeah,
In your research, do you ever find mycotoxins in the biofilm?
I haven't looked, but
Okay,
so I just don't know. I would not be surprised, but I haven't found them.
well, Lyme and mycotoxins seem to really go together.
Yep, I don't disagree at all. But as I
Right.
said, I have focused a lot of my work on Bartonella. I mean, and now consider if we've got more than one organism, we've got mycotoxin, because as I showed in that first picture, biofilms can be very complex. They often have more than one organism. We just haven't looked.
How, how do you see your research translating into treatment?
Well, for one, we are working right now with Dr. Rotello, and we're trying to set up, like I showed you that one experiment where he took cultures of melanoma and cultures of Bartonella, and that's often how drug development works. So we'll work with we're working on a model with him, and we want to see if he can develop designer nanoparticles that will kill the bacteria in a biofilm inside the melanoma cells. So that's we're trying to specifically go after the bacteria. So how it can, well, for one thing it can do with as far as this research is concerned, it tells us that we have a very complex system. It tells us if you just take, I'm sure a lot of you had known this, here, just take the antibiotics for 5 days, you'll be fine. I don't think so. So it's very complex. You need to be retested. And we're looking at different ways to do that quickly. That's what we need to do these quickly. And they're hard. It's hard. It's very hard. And it has not been a very good atmosphere for getting any funds to develop, first to define, to look at them, to look at these organisms, and then to find out where I can tell you, I'm the first one to have found them in a lot of those places where I talked about. People didn't even know. They would say, no, it's not there. Literally, they'll say, no, it's not there. No, you can't find Borrelia. If you don't find a spirochete, it's not there. Well, that's a bunch of hooey. We have dispelled a lot of myths that are out there, and we're hoping with more clarity on what's really there, understanding the biofilms, that we can indeed work on treatment modalities and develop new drugs.
Thanks.
So how does it become apparent on the surface of the skin? Like what would it look like from the outside? Other than the striae
Yeah,
appearances? Do you see any?
Well, I could show you a paper. They just look like a little, oh, you got bit by a mosquito and you just scratched it. There's nothing that's particularly unique about them. That's unfortunate. You can't tell from the outside. The Bartonella tracks, if you're sick, that's one of the things I would definitely use. We're trying to do protocols to use urine, to use blood, to do a skin biopsy, anything we can do, but we kind of got to get inside to see it. You know, Bartonella has been found in mummy teeth that's 4,000 years old. So these organisms have been around a long time to come up with different methods to survive. So we're kind of just starting to pull back the curtain on where these are. So unfortunately, no, if you are feeling ill, and if you have Bartonella bacilliformis, the one in Peru, not a problem. And if you can get fully covered. Some people will get a lesion, but it's often mistaken for something else. So in fact, Bartonella cholerae was discovered by Janet Kohler when she was working with HIV patients in San Francisco, and she found these unusual lesions. And she said, what is this? So she biopsied them and she pursued it, and she found that this was Bartonella, because often Bartonella, like many other infections, they when you are immune suppressed, well, then they'll become more apparent.
Question?
Yeah.
Hi, so I've been on this road for a long time. I'm curious if I used to think of Bartonella as maybe one of the lesser concerns in this panoply of issues that we deal with here. So I'm thinking maybe you want to dispel me of that. And
I do, yes.
And are you in a position to say that Bartonella has maybe now surpassed Borrelia as a major issue of concern, or you're not there yet?
No, we're not there yet. I can say, I think what a lot of researchers are feeling, because I talked about the 3B, that often you're not, because when you get a bite from it, I should have brought that picture, but inside the gut of the tick, it's not just one, it's many organisms.
Yes,
And then they
yes.
get more than one, and I'm sure you know that. And it depends on who you are, what gets put in there, who persists. So we've just been focusing, but we are starting to spend more time with Borrelia, because I think that's very important. And we're looking at Babesia, which is not a bacteria, but it's a protozoan. And so has it surpassed? It hasn't surpassed funding. I usually call these things vector-borne diseases, because some people will say, no, you can't get Bartonella from a tick. Well, yes, you can, especially if you're in Europe. And I just don't think the studies that have been done here to show that you can get Bartonella from a tick have been adequate. And
One more thing,
yeah.
I would like to encourage you, that last slide you have up now,
Yeah.
that everyone dealing with this should see that, because it's very clear you're dealing with something that stays there.
Yeah,
It's
exactly. Yep.
deeply disturbing photo. Thank you.
Sadly, yeah, thank you. Oh, Kathy, I can't hear you. I think you're talking to me.
Yeah. Can you hear me now?
I can,
Hi,
yeah.
this is Erin
Okay,
calling
I
from the
have a
Heirloom
question
Restaurant. I'm just calling to let you know that we do lunch for the rest of this week up until.
Oh dear, somebody send me, okay, never mind, go ahead, sorry. Oh, now I can't hear you again.
Can you hear me now?
Now I can, yep.
Okay. So with Tim being diagnosed with the Rocky Mountain spotted fever, and he has the spinal lesions and lesions in his brain, is that part of what you found, or part of that disease where you would have lesions, spinal lesions? They described his spine as looking like it's been eaten by a moth.
It's horrible, I'm so sorry.
So I just don't know if that's like, you know, what I've read, it says, yeah, you can have lesions, but I just wasn't for sure if it was something. What you're talking about is exterior skin lesions, right? Not
Well,
interior.
oh no, I'm talking about the brain, the heart, lymph nodes, liver, spleen, teeth, everything. I'm not counting out any internal organs at all.
Okay.
But I just think of the skin as the window to get in there initially. And if
So what he has is probably from this tick disease.
It could be,
right?
it could, well, if he has Rocky Mountain spotted fever, right?
Yes.
So he has a different organism, but my opinion, and I am not a physician, so I just can say if it were me, I would get tested for more than one if I was still feeling poorly.
Well, that's what I kind of think, because it says nobody's really ever talked about that. When they sent his blood, they only did Lyme's disease and then this Rocky Mountain spotted fever. They tested for that. Well, Lyme's disease came back negative, but the other one came back positive. So I feel like that we need to have somebody look at other things that could be going on.
So go see Paul, the guy in Kansas.
Yep, I've got his name written down here.
He's a solid guy. He dealt with this a lot, and he understands how this can affect someone, besides being a really smart guy.
Okay.
So yeah.
Well, I appreciate that.
Sure.
Appreciate that referral. So, okay.
Are we,
Are you
anybody
still
else?
doing skin testing, or are people able to be tested if they want to submit some samples, or
Well,
that- because
right
I
now,
kind of thought I heard that
yeah, so right
that
now,
wasn't possible anymore.
well, it is and it isn't. So you can, so we had a funded study from the Cohen Foundation, and we are in the process of, we don't need any more samples, unfortunately. So, but I do work at Tlab, which does take samples, but I don't want this in any way to be about the business. But we look at blood and we look at skin, but so that's all I can say is it's a service that's available. I've worked closely with Dr. Breitschford with Galaxy and other folks throughout the country. So there's a lot of good testing labs around.
Marna, I have a question. Thank you for your slides. This is Dr.
Sure.
Karen Verkota.
Oh, hi, Karen. I know you.
How
Hi.
you doing?
I'm good.
Marvelous slides. And I was wondering, is there any evidence that you found if 23% of healthy blood donors carry Bartonella, then carrying Bartonella isn't the thing that makes us sick. It's something else with the Bartonella. Is there anything else that you found, like if this is there, or markers of inflammation, or C-reactive protein, or anything else that allows, you know, from being a carrier to being a
Exactly.
diseased
Yeah.
person?
Right. That's it. No, that's an excellent question. So I can tell you my colleague in Brazil is actually looking at that right now. So they're looking for, like, say, immune markers, because that's pretty easy, straightforward thing to look for. Because obviously, if they're feeling healthy, they're not very effective. They're not very affected. So we haven't as yet found any markers that would indicate that that you are the one that's going to be sick. We'd love to do that. That would be great. But we haven't yet, but we are looking at it, that's for sure.
Because like with COVID, people with insulin resistance were more likely to get sick and die of COVID. And I would think it would be similar with Bartonella,
Sure.
markers of inflammation, some markers, but
Well, yeah.
I don't know. I was hoping you'd maybe found something next to the Bartonella or something.
Well, but I'll keep, you know what? We are looking. I can tell you we're looking. We found, but I don't have anything where we sort of looked at a population. That's the problem. And I know Paulo is doing that, the guy in Brazil, because he has access to those folks and that blood, because he's at, he can. So yeah, we'd really like to know what else. We have some indication of different markers, but again, it's like one or two people. So I can't say.
All right. Thanks so much.
Love to know.
Dr. Erickson, there's a question in the chat. I don't know if you're
Yeah.
able to see it.
But
Understanding
go ahead.
understanding these findings, any particular treatments, approaches for breaking down the biofilm itself?
That's the tricky one. You find, and I'm sure Karen, you prescribe some biofilm busters, but how do we know they're even, since we've only been able to see biofilms recently, how do we know if those things are even doing anything? We just kind of hope it is. And if you have something to, so I know I've been on them myself, and I'd like to think that they're doing the trick. However, there are some, there's, I take pulmozyme because I have a lung thing, and that breaks up biofilms. That's a known biofilm buster. And acetylcysteine, which is another one that I take for my lungs to break up biofilms in my lungs. That's another known biofilm buster. And so we are looking for different probes that will just show us those biofilms. And we're going to do this in cultures, and we hope to do it in mouse models. So we can find just the long linear DNA in the biofilms, and we can see if those chemicals will break them up. So that and again, I'm not a physician. I only can tell you what I know in the lab. And Karen, do you have any comments on biofilm busters?
You know, I start with lifestyle.
Yeah,
I start
yeah,
with inflammatory lifestyle. And when you start with anti-inflammatory lifestyle, then biofilms don't seem to be able to, everything gets better. And
yeah.
then binders to get the mycotoxins out. And then once you're there, then it is easier to fight the bacteria. But I mean, methylene blue and 660 hertz of red light have been game changers in my practice for
Okay.
Bartonella symptoms.
So there's your answer. There's one known path to go. Boy, would I like to have biopsies from your patients. I guess I show you all this just to give you an idea of how complex it can be. And I think what you're talking about, Karen, with lifestyle changes, it's obviously really important. Really, really important. Are we done? Maybe.
Here's another question from the chat. Are all biofilms bad? Are there any good naturally occurring
There's a
biofilms in
lot
our
of
bodies?
really good ones. Yes, there are. There are many good ones. I think you just have to look into your gut to know that. I worked with cholera at one time and in a mouse model. And what we found with that particular organism, it essentially, the bacteria lines the lining of your gut right on top of all the biofilms. And of course, I think we've shown you want diversity. I have a great picture I could show you about biofilm diversity. And it's a guy in this fellow at the U. He shows a picture of a rainforest, one in the Amazon and one in Southeast Asia. Altogether different plants, but very diverse. And then he shows a field of corn. He says, that's you on antibiotics. You've killed off both the good and the bad. And so, but you have definitely, I again, not knowing that aspect very well, I would say, yes, there are good ones. I think that's pretty well known. Anything else, Britt, online, or is that it?
No, I think that was it. Does anyone else have any questions they want to ask in person? No? Then if you want to turn on your camera and say hello or thank you or good night or anything. Thank you so much for your time, Dr. Ericson. We really appreciate it.
Well, thank you guys. I am so much in favor of you guys having these kind of meetings because we have to get the word out. And if we can do it one by one, that's just fine. Because somebody will hear about it eventually. Or somebody is going to have them or a loved one who's going to get it. And then they're going to become very involved in the research. There's a lot of people that are in the research because of that. Because they've been affected. So anyway, that's what I have to say. So thank you guys. I welcome any questions at any time. So please send any suggestions. Any suggestions? I'm open to anything because we obviously don't have all the answers yet.
Thanks for bringing that information. We really appreciate it.
Sure. Okay. See ya.
Thank you.
Before you sign out, I have one question.
Oh, sure.
And this is a little macabre, and I'm sorry, but you mentioned getting a cadaver.
Yeah.
How would one, and I'm not planning on dying anytime soon.
Okay, good.
If there's an interest in a cadaver of someone who's had Bartonella, babesia, and Lyme, I mean, how would I donate?
Well, there's the Lyme, LLD. There's a group out of, with a bunch of people in New York as well. And there is a LLD. Oh my gosh, hang on. Let's see if I can find it. And actually this cadaver went through them as well. So there is a donation there is a donation website for people with Lyme disease and any others as well. And that would be great. I think anyone who can, if you can make the world a better place, even when you're gone, that would be very helpful. And the brain guys, Brian Fallon in New York, he does a lot of work with that group.
I think at one time Columbia was accepting.
Yes, yep, and Columbia too.
Was it
Yep.
Columbia?
Yep. So we're all kind of, it's all kind of there. There's it's Liz. Oh my gosh, Liz, my friend Liz. She's in charge of getting those cadavers sequestered and sent to the right place.
Thanks.
Yeah. If I find out, I'll let you know via this group.
Perfect.
Good.
Okay. Thank you.
Thank you so much.
Okay.
Thank you.
Have a good night.
Good night.
Thank
Bye.
you all.
Thanks.